And for the next talk, we have Kalli Kappel, who is also from the Broad Institute, telling us about predictive models of the protein sequence and determinants of nuclear condensates. Thank you.
Okay, great. I'm really excited to tell you today a little bit about my work towards predictive models of the protein sequence determinants of nuclear condensates. Got it? Okay, so protein and RNA sequences encode structures that span a range of length scales. So from individual monomers to macromolecular machines to subcellular assemblies. And these structures ultimately control function. Sequence space is absolutely massive, as many people have alluded to at this meeting so far. Even if we consider just currently known protein and RNA sequences, there's about 8 billion of them that we know exist. And I think we'd love to understand what all of them do and how they do it. But this is just a massive problem. And just to give you a sense of scale, if we spent just like an hour characterizing each molecule, it would take us 913,000 years to get through all of them. And so this is really just not feasible. And so this motivates two major challenges. So the first is, can we predict multi-scale structure and function directly from protein and RNA sequences? And if we want to do this, can we generate necessary large-scale data sets that will enable this kind of predictive modeling? And so today I'm actually going to focus mostly on the second question, and I'm going to focus particularly on one type of structure called a condensate. So a condensate is a membraneless assembly that specifically concentrates proteins and or nucleic acids inside of cells. They include really well-known examples such as stress granules and the nucleolus. They're implicated in a wide array of cellular processes, and they're associated with several diseases, especially neurodegenerative diseases and cancer. So what kinds of proteins actually form these structures inside of cells? It turns out that many intrinsically disordered proteins, or proteins that lack a well-defined three-dimensional structure, especially have the tendency to form these types of structures in cells. But not all intrinsically disordered proteins actually form condensates. And so that begs the question, what are the sequence features of these disordered proteins that actually govern condensate formation inside of cells? And so this is a question a lot of people have been really interested in trying to address. And there's been a lot of fantastic work that has gone into trying to address this. I'm showing just some of the studies here. And really just to summarize some of this, it really, depending on what protein you start from and which variants you characterize, people have found that pretty much every amino acid type in some context can be important for driving condensate formation. And so that's kind of an unsatisfying answer. And I think there's really a major challenge here, which is that existing methods measure protein sequences one at a time, one by one, which has really limited scalability. So this has meant that individual studies have focused on a single protein or a family of proteins. Each study has tested up to maybe tens of sequence mutations. And then each study is looking at different sequence perturbations, often in different experimental conditions. And this has made it really challenging to compare and to understand how generalizable these findings actually are. So all of this to say, we lack consistent large-scale data sets. And we really don't have a good way of generating such data sets right now. So with the rest of the talk, I'm going to talk about answering this question: can we develop a scalable method to measure the propensities of libraries of protein sequence variants to form condensates inside of cells? So we developed this approach called CondenSeq to do exactly this. And so I'll walk you through the steps of this now. So we start out by designing a pool of up to about 5,000 rationally designed sequences encoding proteins that we're interested in. So we synthesize these as DNA oligonucleotides in a pooled format. We then clone these into an expression vector that contains a few important components. So first, a barcode sequence, which is just a unique DNA sequence that uniquely identifies each protein. A nuclear localization signal. So right now we're looking specifically at condensate formation within the defined subcellular context of the nucleus. And then a fluorescent protein. In the slides I'm showing you today, this is GFP, which will enable us to visualize the proteins inside of cells. We then create a pool of cells such that every cell expresses one of these protein sequences. Here we're looking at human HeLa cells. And then we can image this entire pool of cells live. And we'll see, this is an example image here. You see a bunch of cells. In some of the cells, you can see a diffuse localization pattern, and in other cells, you see condensates forming. And then because this is in a pooled format, we need to figure out which protein or which cell is expressing which protein sequence. And to do this, we use this in situ sequencing by synthesis technique developed by Paul Blaney's lab at the Broad Institute. So essentially what we do is we synthesize a complementary strand of DNA to this barcode sequence using fluorescently labeled nucleotides, imaging after each nucleotide incorporation. So you see these colorful spots showing up. And in this way, this will tell us which barcode each of these cells is expressing, and therefore which protein sequence they're expressing. We then end up with many, many images of cells for every protein sequence, and we've developed software to then automatically detect and quantify different condensate properties. So now that we have this method, we wanted to ask, across a very wide range of disordered protein sequence contexts, what sequence features really have the biggest impacts on condensate formation. So we designed a library of about 14,000 synthetic sequences to specifically assess the sequence determinants of nuclear condensate formation. So we looked at hundreds of different sequence contexts, and for each of those sequence contexts, we designed hundreds of sequence variants to test impacts of each amino acid, net charge, hydrophobicity, sequence patterning, etc. So suffice it to say, we have lots and lots of data, way more than I can tell you about in 12 minutes. But just to highlight one interesting finding, we found that aromatic and charged residues tend to have the strongest and most consistent effects on nuclear condensate formation across different sequence contexts. So here we're looking at individual amino acid substitutions and their impact on condensate formation. And we can see that the biggest increases come from adding aromatic residues, adding positively charged residues, or removing negatively charged residues. And so just to show this in another way, if we take all of our 14,000 sequences and we bin them by the number of aromatic residues that they contain, and their net charge per residue, and then for each bin, we calculate the average fraction of those sequences that form condensates, where yellow means all of them form condensates, black means none. You can see as we increase the number of aromatic residues, we increase our propensity to form condensates, and similarly, as we increase net charge per residue, we also increase propensity to form condensates, and we can sort of compensate for one with the other. Okay, so aromatic residues makes a lot of sense. It's consistent with a lot of previous studies. Higher net positive charge was a little bit surprising. So in a test tube, you'd imagine that higher net positive charge would cause electrostatic repulsion. But remember, we're performing these experiments inside of cells. And so these proteins have the ability to interact with other cellular factors. And so how do those interactions with other cellular factors actually impact the sequence features that are driving condensate formation? So condensate does not directly tell us about the interactions between test proteins and endogenous cellular factors. But to get some insight into this, we took our library of 14,000 synthetic protein sequences, and we considered potential interactions with all endogenous human disordered regions. So there's about 4,000 of these. And we used this physics-based predictor called Finches, developed by Alex Holhouse's lab, to predict for each one of our synthetic test protein sequences the self-interaction strength, or the homotypic interaction strength, as well as the heterotypic interaction strength, so the interaction of each of these test proteins with every single one of these endogenous human disordered regions. And then what we can do is we can ask for each test protein what is more favorable. Homotypic interactions or self-interactions, or heterotypic or interactions with other cellular factors. We could then take all of our condensate-forming sequences that we identified with CondenSeq and split them into two groups. So the first, where homotypic interactions are favored, and the second, where heterotypic interactions are favored. And then we can ask what's different about the sequences in these two groups. Okay, so I'm showing here a dot plot of a bunch of differences in sequence features between these two groups. Red indicates a sequence feature that's higher when homotypic interactions are favored. Blue indicates a sequence feature that's higher when heterotypic interactions are favored. And I just want to highlight two sequence features. So the first, net charge. Net charge tends to be higher for condensate-forming sequences where heterotypic interactions are favored. So suggesting that higher net positive charge is driving condensate formation through interaction with other factors inside of the cell. On the other hand, if we look at aromatic residues, we see that the number of aromatic residues is higher for condensate-forming sequences where homotypic interactions are favored, suggesting that aromatic residues are more important for condensate formation through self-interaction. So we've used condenseq to do a bunch of other things that I don't have time to tell you about today. We've looked at identifying sequence features that drive co-localization with endogenous condensates. We've mapped the impact of protein valence, looked at roles of chemically similar amino acids, assessed the role of amino acid order and patterning. We've looked at sequence features that drive nuclear concentration buffering, discovered sequence features that control condensate size and number, and analyzed how interactions with endogenous cellular factors can actually tune the sequence features that promote condensate formation. So the next step going forward, we're trying to use our data to learn to predict condensate formation directly from a protein sequence. I won't go into that at all today. I just want to mention briefly that my lab is opening this July at UCLA. We're going to be combining these kinds of high throughput experiments with computation to decipher these multi-scale sequence structure function relationships of proteins, RNA, and their interactions. So if you're interested, please reach out. And with that, I'd like to say a huge thank you to my advisors, Aviv Regev and Feng Zhang, and to all of the lab members who have been really helpful throughout this work. And thank you.
Hi, I'm curious, did you try any of your test proteins in the cytoplasm rather than the nucleus, and how important is that localization to what happens?
Yeah, it's a great question. So we haven't done that yet. Obviously, it's like one of the sort of obvious next directions for this. I would expect there probably are some sequence differences between the nucleus and the cytoplasm. People have shown that actually a lot of times pathogenic mutations in proteins that cause mislocalization between the nucleus and the cytoplasm can actually cause aberrant condensate formation. So I would expect there probably are differences, but we haven't looked into it yet.
That sort of leads into the second question, which is, can you distinguish between some kind of specific condensation versus just protein aggregation, like the hydrophobic amino acid just being really sticky?
Yeah, so you mean like whether we're bringing together like specific collections of other molecules inside of the cell, or maybe I don't exactly.
Whether this is a specific protein interaction or whether this is just non-specific positive charges binding to nucleic acids, right, or hydrophobic interactions amongst aromatics.
Yeah. Okay, so we definitely think that, for example, nucleic acids are very important in condensate formation. So the net positive charge, these things are likely interacting with nucleic acids. So we looked at, for example, things that localize to the nucleolus and chromatin, and we see very high enrichment of net positive charge, suggesting that nucleic acids are indeed very important for condensate formation there. But yeah, one of the huge future directions is trying to figure out like what else is inside of these condensates. And I think some of these physics-based predictors are particularly powerful for starting to get at that question at the scale of sequence space that we've been looking at.
My question is, do you think this condensate, the mechanism principle you find is a cell type specific, or is a general principle that you are extracting from the screen?
Yeah, it's a great question. So we've only looked in 2 cell types so far. We haven't seen any huge differences, but I would expect that it should be cell type specific, at least to some extent, right? So we've done a bunch of analysis looking at interaction, like predicted interactions of these test proteins with the endogenous proteome. And we see that depending on what predicted interactions they make, different sequence features are important for driving condensate formation. And so that tells me that if we have different cell types where different collections of proteins are actually being expressed, that you would expect that there is some differences in what is forming condensates.
Now that you find the principle about the aromatic uncharged residues, for example, have you been able to do some loss of function, either simulation in silico or do experimental validation in some endogenous protein? You mutate, you remove those residues. Now they can no longer.
Yeah, so we've looked at we've looked at like longer segments of real proteins, yes, and we've seen that if you make these kinds of mutations, you do see, like, if you remove aromatic residues, you can remove condensate formation. There's also a lot of orthogonal studies that have been done in the field that validate a lot of the findings that we've had.
Maybe you could also scale it up. I'm thinking about AlphaFold3, like multimer, like a structural level. Can you predict the dimer formation disrupted by this mutation?
Yeah, so it's a good question. So I mean, we're looking at disordered proteins right now. So I think you have to think a little bit differently about the way these things interact. Of course, some of these disordered proteins are likely folding up and binding to things, but a lot of them, we know that disordered proteins can bind specifically to each other, but not form traditional sort of like ordered protein-protein complexes. So I think we have to think about it in a little bit of a different way, yeah.
All right, we have a question from Katie Farrick on Zoom. Have you looked at predicted phosphorylation sites or other post-translational modifications and what role they might be playing in condensate formation, assuming some of your test proteins may be modified this way?
Yeah, it's a good question. So we've done a couple things. We've looked at phosphomimetic mutations, and you see lots of impacts as you would expect. So in many cases, having a phosphomimetic mutation will decrease condensate formation, but not in all cases. So there is some sort of like sequence dependence to this. And then we've looked at like subsets of these proteins that don't contain any phosphorylatable or any commonly phosphorylatable residues, and we see that we see the same sort of sequence trends. So we don't think that like the net positive charge, for example, is due to things being phosphorylated inside of cells. But yeah, this is something again that would be interesting to follow up in more detail on.
Quick last question.
Are you able to determine the impact of the cell cycle from these snapshots?
Ooh, yeah. So we have, in theory, I think yes. We haven't looked at it yet. Yeah, but it's a great question. Yeah.
Okay, let's thank Kalli again.